Uh, Today we'll see about this apparatus.
Yes.
This apparatus is the traveling microscope.
Okay.
Traveling microscope is a measuring device. It measures the size of the small objects. So it is made up of one microscope, and this microscope is having the ability to move. So let's see the different parts. This part is the microscope. So this is traveling microscope. We can adjust this. So the microscope can be adjusted like this, and with the help of this screw it can be fixed. Next, we can use any object to see, and this adjustment is called focusing adjustment. We have to adjust the distance of the microscope from this object in a proper way so that we should get the clear image. So once the image is obtained clearly, we have to fix. After that, we have to note the location of this object. In order to find the location, there are the two scales. This is horizontal scale, and microscope can move on this horizontal scale. This is the horizontal scale. In this horizontal scale, this particular part is called the main scale. Main scale is graded in centimeters. So here the marks are there which is indicating the centimeters right from 0 to 20. So these are centimeters. This is called the main scale, and over this main scale the microscope is movable. Microscope is movable on horizontal scale, and along with this microscope there is a little scale. This scale is called the vernier scale. Vernier scale has 50 divisions starting from 0 to 50. Totally 50 lines are there. This microscope is able to measure the size of the object to the accuracy of 0.001 centimeter. So the least count of the apparatus is 0.001 centimeter. That means it is able to measure 1/1000 part of the centimeter. Also able to move along the vertical scale also. So this is the vertical scale, and on this vertical scale the microscope can move. So here on this vertical scale also some grades are there. This particular part is called the main scale, and on this main scale the gradings are there right from 0 to 15 centimeters are there. And over this main scale this microscope is movable, and along with this microscope this movable part is there. This movable part is called the vernier scale. Here again the vernier scale has 50 marks, 50 divisions are there on the vernier scale. So this vernier scale is also able to measure the location of the object to the accuracy of 0.001 centimeter, or 1 centimeter is divided into 1000 divisions. So this microscope is having the ability to find the location of any object to the accuracy of 1/1000 part of centimeter, so that the least count of the apparatus is 0.001 centimeter. Here one thing the student has to note, that the measuring device is having the accuracy or least count of 0.001 centimeter. Means the accuracy or the least count to be expressed in the units of centimeter, and moreover the digits should contain one decimal point. After that 3 digits will be there. For example, the least count is 0.001. Now let's see how this apparatus is used. There are the different controls. This control is called the focusing adjustment. So microscope is used and the object is placed at this place. After that the microscope is adjusted for its clarity. So focusing is adjusted. If proper focusing is not obtained, it is possible to use this screw. This screw is used for adjustment. So microscope can move when the screw is released. Once the image is obtained with clarity, the screw is fixed. After fixing the screw, this control is used for further clarity. When the image is obtained with much clarity, then this is released. After that, the location of the image is noted on, for example, the horizontal scale. The reading of horizontal scale is taken like this. In order to find the location of the object in digital form, we have to see the vernier scale. In this vernier scale, we have to locate the 0th line. For example, here the 0th line is lying after 4 centimeters on the main scale and before 5 centimeter. The exact location of this 0th line, the 0th line is that line which is available on the vernier scale. That 0th line is found to be after 4.5 centimeter. That means on the main scale, the reading of 4.5 centimeters have been crossed. So the main scale reading is 4.5. Moreover, on the main scale, each millimeter is again divided into 2 halves. So the exact value is 4.5. So 4.5 is the main scale reading. After noting this main scale reading, we have to find the extra part in the vernier scale. Out of 50 lines available in main scale, we have to see which line out of the 50 is meeting, coinciding exactly with the main scale. For instance, here the line number 24, is coinciding with one of the main scale reading. So vernier scale coincidence is 24. Now this 24, line number 24, is multiplied with the least count. The least count is 0.001 centimeter. So this extra part becomes 0.024 centimeters. Now this extra part is added with the measurement counted in the main scale. So main scale reading has obtained as 4.5 centimeters. This 4.5 centimeters added with 0.24, the total answer becomes 4.524. 4.524 is the total reading. This 4.524 centimeters is the position of the object on the horizontal scale. Now we can move this microscope on horizontal scale. For the purpose of movement of that, we have to use this control. This control is called vertical movement control. When this vertical movement control is fixed then the chorus control will work. And now the fine control, this particular part is called the fine movement along the horizontal scale. If we operate this, the microscope moves slightly on horizontal scale. Thereby we can change the location of the microscope. So microscope used to move like this. When microscope is moved to the other side of the object, then we have to see in the image. The image has gone to the other end of the object. Now, when we stop here, we have to take the second reading. We will get a different second reading. So the second reading is also obtained the same way as the first reading is obtained. Now, if we take the difference of these two readings, we will get the total length, the extent to which the microscope has moved. The extent to which the microscope has moved, that is going to give you the size of the object on horizontal scale. In a similar way, the height of the object along the vertical axis is also be obtained by noting the 2 times the reading. So difference of the 2 readings will give the size of the object on horizontal scale. So this is the way we can use this travelling microscope.